Hola, buenos días a todos. Bienvenidos a el último seminario del año. Y el día de hoy eh, me da mucho gusto presentar a la ponente del seminario, que es la doctora Susana Rocha. La doctora Susana Rocha hizo su doctorado y su postdoctorado en la Universidad Católica de Lovaina y bueno, durante su carrera ha tenido muchas contribuciones tanto en el área biológica y últimamente en el área de materiales. Tiene muchísima experiencia con la parte de microscopía óptica de fluorescencia de super resolución. Entonces, me parece que es un área muy importante y que en el CIO está cobrando más y más relevancia cada vez. Entonces, sin más preámbulos, please, Susana, I give you the word. Thank you very much for accepting the invitation.
Thank you for inviting me. So if you have not guessed from my name, I'm from Portugal, which means that I can understand Spanish. So what Eduardo was saying, I could understand. But unfortunately, I cannot speak Spanish. I will not even try so that we don't embarrass you or me. And I'll just do the presentation in English. So when Eduardo asked me if I was interested in giving a seminar, he told me that people in your research group or environment or institute were interested in super resolution, and you were from a variety of backgrounds. So I wanted to talk to you a little bit about the super resolution that we do in the lab, but that is mainly applied, at least so far, to biological systems. And then because we also work with materials, and I know that there are probably more of you working with materials also, then I put a part of using fluorescence microscopy to study materials, and in this particular case it will be hydrogels. So we'll go first for super resolution and then continue to the materials. And when we talk about super resolution, when I have to teach it in class, I always like to show this slide, because at least during my primary school, maybe high school, we all talk about the light and how it diffuses. And if you have a slit in a paper or in a wall, that the light, because it's a wave, it will create this diffraction pattern when it passes through this thin slit. But if that hole, instead of being just a rectangle, is just a circle, this is then the image that you get. You don't get these lines pattern, but you get a circular pattern that is then an airy disk. And what happens when you have an objective and you are focusing the light is something very similar. Your light cannot be focused in a single point. It will always show this diffraction pattern. And this has consequences for optical imaging, because if I show you a typical image of fluorescent molecules, in this case, they are single molecules, but they could also be, for instance, viral particles, or they could be protein aggregates. If you see all the different dots, they all have a certain size, and here each pixel is around 100 nanometers. So even though these are fluorescent proteins, which are approximate 5 nanometers, they show up as 200 to 300 nanometers. And that's because the light has this wavy characteristic, and it creates a diffraction pattern. So when you look at these images, you cannot know what is there. You cannot know the real size of the object that you are imaging. It's a diffraction limited image, which means that everything below 250 nanometers will look exactly the same. Either it is a single molecule or a virus or a protein complex. And this has consequences for when you are imaging more complex structures. So if you have different fluorophores arranged on your sample on a different or specific arrangement on the pattern, because every fluorophore will show up as a diffraction limited spot, which is much bigger than the original molecule, instead of seeing the arrangement of your molecules or your fluorophores on your sample, you see a blurred image that we usually call a diffraction limited image. And what is this? I would say, how sharp is your image? The diffraction limited, the resolution of your image is defined as the once you have the two airy discs, when they come too close and you are no longer able to discriminate them. So here you have two patterns that are very far apart. Here they are close together, but you can still clearly see that they are two different fluorophores. But once they become closer than a certain distance, you cannot resolve them anymore, and you start having then blurriness, or you are unable to distinguish the two objects. And that distance is related to the wavelength of the light and to the numerical aperture of your objective. Usually with the numerical apertures of current objectives, we are talking about a resolution of around 200 to 300 nanometers typically. There are different ways where it is possible to overcome or to circumvent this diffraction limited on the microscopes, on the light microscopes. There are several methods. Here are just the three most used or most commonly used methods that are based on STET, structural illumination, SIM, and then localization microscopy. And a few years ago, some people in the lab wrote this nice review paper that was really meant for people that come from biology, that have no knowledge on optics. But Edward just told me that you all have knowledge on optics, so maybe this is not the perfect review for you guys. But when a student joins the lab and wants to work on super resolution, this is always the article that we tell them to read, because it explains everything from very basic concepts in optics until then the more specific concepts related to these three super resolution microscopies. I work mainly with localization microscopy. We do have a STET microscope. It's a commercial STET microscope at this moment, and it also works, but there are still some limitations in terms of having the resolution that is claimed in articles. We can get around 50 nanometers if we are lucky, but we can never reach the 10 nanometers that are claimed in some publications. And with the localization microscopy, we can actually reach easily 20, 25 nanometers. So we tend to use that more just because we are more familiar with it. And the SIM, we actually don't even have a SIM in our department. There is one in KU Leuven that we sometimes go to, but I think there is a home-built SIM, but the resolution was also not as good. So we just use, or I use mainly localization microscopy. And how does this work? So if you have your fluorophores and they are arranged on your sample, because of the diffraction limit, you just see this blur pattern, and it's very hard if you look at the final image that you have, it's impossible to know where each molecule is. But if you have this image on your camera, and if I tell you that this is only one molecule, if you are 100% sure that this is only one molecule, then you know exactly where the molecule is. I think it's very easy to assume that the molecule is in the center. If you only have one molecule and you have this signal, the molecule will not be in the corner, right? So the molecule has to be here. So if it's only one molecule, you can know the position of this molecule. And this is done by simply fitting the signal that you get to a 2D Gaussian. So this 2D Gaussian is just an approximation of the airy disk pattern that you get. And by fitting the point spread function that you get on your detector, you can calculate the position of your fluorophore with a certain accuracy. And this is just an equation that just tells you how accurately can you determine the position of the molecule. And this depends, of course, on the number of photons. So the more photons you have, the less is the error on your calculation, which means the more precise you can determine the molecule, which makes sense, because if you are getting more photons, your signal will be sharper and it will be easy to know where the center is. And also, of course, the background. The more the background, the more noise you have, and the worse is your determination. But what you really need to be 100% sure when you are working with localization microscopy is that you only have one molecule per diffraction limited area. And to do that, you have to be able to control the fluorophores. You have to be able to turn them on and off. And imagine that you have that power of turning the fluorophores on and off. If you have your sample, you can turn on a few fluorophores, because you know that in each diffraction limited pattern there is only one, you can determine the position of the single fluorophore with a high accuracy, and then you turn the fluorophores off, you turn a new subset of fluorophores on, you determine the position, and you keep on going until that eventually you have imaged all the fluorophores in your sample, or at least you have imaged enough fluorophores that allows you to reconstruct the image that will then correspond to your sample. So localization microscopy is a technique that really needs to have single molecules on the diffraction limited spot, and then you reconstruct your image by determining the position of the molecules one by one. Okay. The trick is not on optics. It's just a very simple wide field microscope. As long as you can see single molecules, you can do localization microscopy, palm and storm. The optics is very easy. The advancement or the essence of this technique is related to the fluorophores that you use. And there are different ways where you can turn fluorophores on and off, that you see them at certain points and at other points you don't see them. One of those ways is to use what we call the smart labels. So these are molecules that have two states, either a dark and a bright state, or two states with different colors, so that if you only look, in this case, if you are only looking to the yellow channel, this molecule converts between green and yellow, but actually if you are only looking at the yellow, it looks like an on and off behavior. Or you have these molecules that you can turn between two states, and those can be fluorescent proteins or organic dyes. Or you have molecules that interact with your structure of interest, with your sample, in a reversible manner. So imagine that you want to image this star here. You just need to have a molecule that interacts with this star. So you can imagine that this star is an actin filament, and you have a protein that interacts with the actin filament. And then every time this protein comes and attaches to your star, it will give a signal. And this signal you can detect. This corresponds a little bit to your own state. And then when the molecule detaches, because it needs to be a reversible binding, then it's off again. So in this case, if you have smart labels, you control the on and off behavior by changing the molecules between two states. And in the case of reversible binding, you have an on and off behavior because the molecules bind and unbind to the object that you want to image. So far, actually, we use a little bit both, but what I'm going to talk to you mainly about uses these smart labels, and it uses fluorescent proteins. So fluorescent proteins, they got in 2008 the Nobel Prize for the EGFP, but there are many more fluorescent proteins. I always like to show this slide because we call it the fruit series. So you have all types of fruits have a name in a fluorescent protein, but there are many more. And if you are ever interested, if you have a fluorescent protein and you don't know the properties of the protein, there is this very nice website that is a fluorescent protein base, and it has 945 fluorescent protein spectra. So if you just type in the name of fluorescent protein, you have all the information about it. There are a lot of fluorescent proteins, but we are not interested in all fluorescent proteins. We are interested in a specific group of fluorescent proteins. We are interested in proteins that switch between two states. And then there are different types of classes within these photo-switchable fluorescent proteins that are the ones that are photoactivable, or they are dark, and then once you irradiate light, they become fluorescent, so they are photoactivated fluorescent proteins. You have the ones that have one color, and then once you irradiate with UV light, they change color, the structure of the chromophore changes. And then you also have the ones that you can switch them on with the laser light and then switch them off with using a different wavelength. And for every category, I just wrote here the name of a few fluorescent proteins that are commonly used in literature for palm, super resolution localization microscopy. the PA GFP, PAM cherry, PA tag RFP. So the PA stands for photoactivatable. And then you have the ones that switch Kaede, MAOs, and Dendra. Actually, MAOs, there is a series of MAOs. There is MAOs, MAOs 2, MAOs 3.1, 3.2, MAOs 4, I think, by now. And then you have the ones that switch. You have a DROMPA and a reversible GFP, just to give an example. How does it work in real life? Here is an example of some cells. So sorry, there is missing the scale bar, but this is around 50 micrometers by 50 micrometers. And this is the plasma membrane of the cells. So we are measuring in TIFF, which means that we are only imaging the plasma membrane of the cells. And the cells are expressing a photoconvertible fluorescent protein, MAOs 3.2, target to a small lipid sequence that makes the MAOs 3.2 be at the plasma membrane. And you see that the protein is not uniformly distributed through the cell membrane. You have some kind of structures within the cell membrane. And this is the diffraction limited image. The MAOs protein is photoconvertible, which means that it is originally in a green state and it's expressed everywhere. So if you just look at the green channel, you see a diffraction limited of the distribution of this protein. But if you look at the red channel, and if you put a little bit of UV light, you'll see an image like this, just a few dots, okay? So every one of these dots is a fluorescent protein that is on the red state. And the reason why these look so much different than that is because in the case of the dots, you only have a small subset of the proteins that are on this red state. And then, wait, let me try to play this video. Should work. Go back. Yes. So, and then if you just acquire a data set, you see this kind of blinking. So what happens is that in every frame, there is this continuous, very weak illumination with ultraviolet. And every frame, there is a different subset of molecules that are converted from the green to the red. So they are converted, and then they are imaged with 560 nanometers excitation, and then they bleach, or they go to a dark state on the red. So you see them for a few milliseconds, and then they are off again. So you have this on and off behavior, and every time you are activating different molecules. If then you go through your movie, and for every frame, you fit these dots with the 2D Gaussian, and then you know the exact position of every molecule. And then you reconstruct the image, so you plot for every position that you acquire during the movie. And usually we do 5,000 frames, so it takes around 4 minutes to acquire this data. Then you get images like this. And then you can see that even though from the single dots you are not able to discriminate the structure, if you acquire long enough, if you acquire enough single molecules, you can kind of get the same image as a diffraction limited, but of course with an improved resolution. So here is just a zoom in. This is just a zoom in of the same area. And for instance, here you see what I call the PlayStation 3 remote control feature on the diffraction limited, and then you see it on the super resolution. And I think you can agree that the resolution is much increased. Also, like here, you don't see that it has two edges, and on the super resolution image, you really see that. So this we do with fluorescent proteins that can convert between the two states. We can also do it with organic dyes. And the method more used to do super resolution localization microscopy with organic dyes is called the D-storm. And the whole idea with the D-storm is that you add into your solution a buffer that contains a reducing agent and an oxygen scavenger system, so that you reduce the concentration of your oxygen. And when you do that, usually you have your molecule, you are irradiating it, it goes between the singlet state, the ground state, and the excited state. Eventually, some molecule will end up on the triplet state. And what this special buffer does is that it offers an alternative pathway for the molecule on the triple state that makes it go back to the singlet ground state with a higher probability. And because it induces this alternative route, the molecule can blink on and off several times before it's completely bleached. And this is done mainly with Alexa 647, but there are also different dyes. This article is already from 2009. Now there are many more where you can, by using a special buffer, you can tweak this on and on behavior of the dyes. And I have to say that even though in literature people talk about PALM as mainly for fluorescent proteins, and they talk about STORM as for organic dyes, the two are a little bit interconnected. And it's at the end, it's just single molecule localization, and which dye you use, it doesn't matter. And we showed in the past that we could use this to study the structure of HIV. So now everybody's familiar with a different type of virus, but when we were doing these studies, HIV was the hype virus at the moment. So in HIV, what you have is that you have your lipid bilayer. Outside of the lipid bilayer, you have these envelope proteins that work to recognize the receptors on the cells. And then you have your matrix protein that forms the inside of the lipid bilayer. And then you have the capsid. And inside the capsid, you have different proteins like the integrase, the proteinase, the reverse transcriptase. Those are all inside the capsid. And we know that the structure of the virus is like this from electron microscopy images. But when we put them on the microscope and we label two different components of the HIV particles, you actually see that they all look the same, just diffraction limited spots. But if we then do super resolution, and here we labeled different proteins with either a fluorescent protein or with antibody that was then coupled with an organic dye. And we can see that this protein that is supposed to be on the core of the virus shows a very narrow distribution of sizes. The integrase is also very small, so it's also inside the viral core. And then the capsid is a little bit bigger. The matrix is again a little bit bigger. And then we were expecting to see the envelope also bigger, just a more blurred structure. But then we start getting these Mickey Mouse kind of structures, and we were like, what is this? Something wrong with antibody labeling? But then we look back into literature and into some EM images, and actually it seems that these envelope proteins, they form clusters on the surface of the HIV particles, and that's what we were seeing when we were imaging the viral particles. You had this, I'll say it again, Mickey Mouse. So these dots around the virus particle and the number of envelope clusters were just actually in good correlation with the electron microscopy. And this was just a very basic study that we did back in 2008 or something. And then from there until now, we used super resolution on different topics. And the most recent topic that we are working on is on Cluttering organization. So Cluttering it's a molecule that you have the heavy chain and the light chain, and it actually forms these structures on the plasma membranes that are then responsible for clathrin-mediated endocytosis. So basically, when cell takes up something in, it can be through the formation of a clathrin vesicle, and basically the clathrin assembles around the content of the vesicle, forming this clathrin-coated vesicle. But when you look at images, electron microscopy images of clathrin, you see indeed these vesicles that we call the classical clathrin-coated structures. They are around 100 nanometers, very spherical. But on the electron microscopy image, you also see these arrays of clathrin organization that do not form a vesicle. And literature describes this as flat clathrin lattices. And it's still a little bit unsure what is their main purpose. They also form vesicles. So you see it here, for instance, there's a vesicle being formed from the edge of this plate. But further, what is their real function, nobody really knows. There is some hypothesis that is related to cellular adhesion, but there is not a lot of evidence to support that. So what we wanted, there is some information, sorry. These flat clathrin lattices have been detected in several cell lines, and mainly in cancer cell lines. And we also know, some problem here with the squares. We also know that these cancer cell lines, that they overexpress specific proteins. And in the case of cancer metastasis, so when the cancer is leaving the primary tumor to go to a secondary location, it was found that the cells that have this potential to create metastasis, to go to a secondary location, they express or overexpress proteins that are involved in the clathrin-mediated endocytosis, namely AP2 and dynamine. So we start wondering, okay, is there a link between how the clathrin molecules are distributed to the metastastic potential of certain cells? And so we picked up colorectal cancer, and we took two cell models, the KM12 model and the SW cell model. So you have the cells that come from the primary tumor, from the colorectal cancer, and then they inject those cells into mice, in the case of the KM12, and they develop a secondary tumor in the liver, or in the liver and in the lung. So these cells come from the liver or from the lung. And then the SW model, it was actually from a patient that developed cancer on the lymph nodes. Okay, so you have these cells that have the same genetic material, the same DNA, but they just have different metastatic potential. And we start looking at clathrin organization within these cells. And this is what we start seeing. So this is just confocal imaging, very boring normal microscopy. So here is the non-metastatic cell, so the good cancer, let's call it like that. And you see that there are a lot of small dots. But once we start going to the cells that have a higher metastatic potential, you start seeing these very extensive structures of cluttering. And we don't really knew what this was. Was this just aggregation of clathrin pits, or are these really these flat clathrin lattices that have been reported in literature? And because it is diffraction limited, we could not be sure. So what we did is that we did super resolution. And here you have the same cell in confocal imaging and then in palm TIRF imaging. And you can see two things. A, the resolution is better. So you can see here, for instance, these three dots, while here it just looks more like a line. So here you can clearly discriminate three spherical structures. But then you also see that there is much more background on the confocal image than on the palm TIRF images. And that is not related to the super resolution per se. It's just related to the fact that we are measuring in TIRF. And as a consequence, we only really image the clathrin that is on the plasma membrane. And on confocal, even though you have some Z sectioning, you still have around 600 nanometers that you image. And as a consequence, you get some of the clathrin that is just above the membrane being endocytosed. So some of these vesicles that show up on the confocal image, they are not present on the super resolution imaging. So we saw, okay, these structures, even though some of them are just like pits, some of them are really these long structures. So can we quantify these? And then because with super resolution, you have the localization of every single dot that appears on your image, we can do some more sophisticated analysis. And we use a clustering algorithm that is called DB-scan, that from all the localizations of the different molecules, it just calculates, if you give the right set of parameters, it calculates clusters. Okay, so here each color corresponds to a different cluster. And then what we did after was saying, okay, if we assume that clusters that have a very big area, a lot of molecules that are not spherical, that are a little bit elongated and have a long perimeter and are close to each other, we say, okay, those are a flat clathrin lattice. So this extensive structures. And then we can look back to the image, and to each cluster that was identified, we can say, okay, these ones are the pits, the spherical cluttering structures that are detected in endocytosis, and then you have the pink ones that are these extensive cluttering lattices. And then we image a lot of cells, and here are examples of two different cell types, and it's a little bit of a test for you and also for me. If you look at these ones, you see mainly small dots. You also see these more broad, extensive arrays, like for instance here or this one, that it also looks like a lattice, a cluttering lattice. But then if you have this other cell, you see that these extensive cluttering structures are much more dominant. You can find them much easier. And then we just say, okay, is there a relation between then the metastatic potential of the cell and these cluttering structures? And in fact, there is. So if the cells are non-metastatic, either the KM12 model or the SW model, you can see mainly the pits, the spherical structures. But then if they have a higher metastatic potential, you can start seeing these flat cluttering lattices. And we can see, if we then analyze everything, we just calculate the ratio between the cluttering pits, the spherical structures, and the lattices, which are these more elongated arrays. And we see that the non-metastatic, this ratio is very low. And when the metastatic potential increases, the ratio increases also. So that means that there are more and more of these flat cluttering lattices in comparing with the non-metastatic. According to the reports on literature that these structures might be involved in cellular adhesion, we think that they might have a role when the cancer is spreading, in having the cells then being attached at the secondary localization. But yeah, that we still need to investigate. But I think it's a nice example to see how we can use super resolution to quantify things in cell biology and to have some insights. And just, I had some slides, but I'll just skip that because it's not so important. And I'll just go to the second part of the talk, which is then the material characterization and how we can use fluorescent microscopy to work on material characterization. And as Eduardo said, I just started working with this. And our interest was, we had some hydrogels and we want to characterize these hydrogels. But if you look into the literature, the techniques that they are to characterize hydrogel are quite static, right? You can do electron microscopy, you can do some rheology, which you get dynamics, but it's from bulk. And then you can do also some Sachs measurements. If you want to calculate things more locally, like for instance, the stiffness of the material, you can use AFM, but then you are just really probing the surface. So we thought, okay, actually, fluorescence microscopy has been so used in biological sciences that in the material sciences, and especially in the soft material sciences, not related to photonics or anything like that, there's really not a lot. So we really thought that fluorescence microscopy can actually provide an alternative way to characterize materials on a microscale level, which is often important for biology, and in a dynamic way. So if cells are interacting with the environment, that we can probe this interaction. And so we set ourselves on the path to develop tools based on fluorescent microscopy that were able to characterize both the structure and the mechanical properties of the hydrogels. And I have to say that I was very optimistic when I was making the presentation, but if I look at the time, I think we'll just talk about structure. The mechanical properties using microrheology is very, very preliminary, so I'll skip that part at the end. Which hydrogels are we using? There are different types of hydrogels, and often in literature they are classified as natural hydrogels or synthetic hydrogels. So natural hydrogels are based in proteins or natural occurring components like sugars. And usually, especially if one speaks about protein-based natural hydrogels, they have a structure. So if you talk about collagen hydrogels, collagen molecules, they first assemble into a bundle, then they into a fiber, then into a bundle, and then the bundles form this network. So there is a kind of a hierarchy, and they are very dynamic systems. They are used to respond to changes in the environment, mainly induced by the cells, and they have this fibrous structure, very porous structure. On the other hand, you have the synthetic materials, and those are typically chemically cross-link polymer materials. So polyethylene glycol is used a lot. Polyacrylamide hydrogels are also used. So it's you have your monomer, and then you induce the polymerization, and you have your material. But as a consequence, there is no hierarchy, everything is the same, and your pores are much smaller, and your fibers are just basically a monomer molecule, so they are not very thick. And in the last 5 to 10 years, in terms of biological applications, there's been a lot of interest in developing supramolecular materials. So these are hydrogels that are formed due to non-covalent interactions, so physical interactions. And why are people interested in that? Because the fact that there is a physical interaction makes these materials more dynamic, and therefore it's more interesting if you want to add them with cells, because then the cells can modify the material to their demand. And in the lab, we use this material that are called polyisocyanopeptides, or the PIC hydrogels. And it's a fully synthetic material, but it's quite unique because during the synthesis, it forms this backbone, forms the helical structure. And because of the hydrogen bonds between the side chains, this helical structure remains after the synthesis. And this renders the polymer a semi-flexible character. So a normal polymer in solution just coils up and forms a sphere of coiled polymer. But because of this helical backbone, the PIC hydrogels, they don't form this coil. They are semi-flexible, and they have a very nice characteristic. They form a gel when you warm up the solution. So it's an entropy-driven gelation. It's physical interactions between the side chains, but if you have it at below 18 degrees, it is a liquid, and once you warm it up, it forms a gel, and it's fully reversible. And the process behind this gel formation is really a formation of polymer bundles. So the side chains, the ethylene glycol side chains, once you start warming up, they become hydrophobic, and they are attracted to each other, and they form these bundles. And this type of fiber structure is very similar to what is observed in collagen and fibrin. But then how does this fiber structure actually look like? And when we start working with this material, there were a few studies focused on trying to understand or to know the structure of these PIC hydrogels. There was some work with AFM, and in that case, they just put a solution of the hydrogel in a microsurface, they dry it, and then they check what type of structure they have. So this is a complete dried sample. Then there is also cryo-EM, where again you have a fixed sample. Here you can really nicely see a nice porous structure. So here you have the scale bar. This is 1 micrometer, so they are very big pores. But because of the way as the image is acquired, you just have like a 2D projection. So it's really hard to know how big are the pores actually. And then in SACs, you actually look at the small pores, and then you also see that there are differences when you change different polymers, but you are only looking at a very small nanometer scale. You cannot see it on the micrometer scale. Because it is a fully synthetic material, we are able to label it with a fluorescent molecule. So what we did was that on the side chain, we add the fluorophore, and then we just acquire some fluorescence microscopy. That's what we do. So this is just a Z plane of hydrogel that is labeled with the fluorophore. And then you see here the yellow are all the fibers, okay, and the black are the pores. And again, I'm keep forgetting to put the scale bars, but from here to here it will be around 20 micrometers, okay? These pores will be like 4 micrometers. They are very big pores. And then what we do is that we do some analysis on these pores. So we just segment it based on intensity, and then we can kind of do this ball filling model, so that we try to fit the pores with some spheres to have an idea of how big are these pores. And because we acquire the data in 3D, you can actually calculate the pore size in 3D, and we can calculate the connectivity, so we can see how porous is the material, how connected are the pores. And then we did that for different concentrations, and we see nothing very spectacular there, that once you reduce the concentration, so you go from 1 milligram per ml to 0.5 to 0.25 milligrams per ml, the pore size increases. But even with this very low concentration, with this huge pore size, the material is still a gel, and it can be used to grow cells, but I haven't put any of that data here. Then we are also thinking, okay, we now know how to quantify the structure of this material, but we can take advantage of the fluorescent microscopy to do it in a dynamic way. So we mixed the PIC hydrogels with polynipan. Polynipan is the material that contracts once you warm up, and we were expecting it to change the structure of the hydrogel because the mechanics of the gel changes a lot. So here you see the stiffness of the material once you increase the temperature. If you have the PIC only, here is basically a liquid. Here it starts the gelation of the PIC, so it starts forming a gel. And then, yeah, as you increase the temperature, it just increases a little bit the stiffness. Oh, sorry, here. No, the red one, yes. The polynipan, you see, it collapses at around 32 degrees, the polynipan that we are using. So above 42 degrees, the stiffness increases. And when you mix the two systems, you have the PIC gelation, fine. And once you have the collapse of the polynipan, it also increases the stiffness. So there is a change in terms of the mechanical properties, and we wanted to know if there was also a change in terms of the structure. We label the polynipan and we label the PIC. So the PIC is similar to what we had seen before. The polynipan is not a polymer, it's just the monomer that we added. It's distributed through the volume very uniformly, so we don't really see any structure. But once we warm it up, this is what we see. The PIC hydrogel, you can see the difference. The fibers become more compact, more compressed, I would say almost, and the polynipan kind of collapses on top of the PIC. So we think that's because the polynipan becomes more hydrophobic, and the PIC fibers are also hydrophobic, so they kind of collapse together. And we even have a movie where we warm up the sample. I hope you see the movie. And then you see that it goes from being just very not diffuse, but a broad fiber, and then once you warm it up, the polynipan compresses on the PIC, and you see the PIC fibers become like brighter and more compact. So then we are now trying to quantify the structural changes using some analysis. And then another thing that we also still did, that I still want to show to you, is to play a little bit with the solution. So if you have PIC only, you have here the gelation temperature is around 18 degrees. But if you add to the solution some salt, you can nicely change this gelation temperature. You can go to lower values or to higher values. And if you look at the mechanical properties, you would say that's the only thing that is happening when we add salts. You are just changing the gelation temperature. But actually, when we look at the microscope, it looks like the structure of the material is also different. So here you have the normal PIC just in water, and then you have here the PIC with 1 molar of sodium perchloride, and you see that the structure is completely different. The fibers are much brighter, much more dense. And one thing that we really wanted to do was to follow the gelation process, but we could not do it if it's at 18 degrees, because then you have to cool down from room temperature, and we didn't have that possibility. But if we add the salts, we can actually warm up, because we moved the gelation temperature to a higher temperature, and then we could follow So here we can follow how the gel forms, and actually it looks almost like an instantaneous process, right? So there is nothing there, and then everything is formed. I'll just play it again. So you have a very uniform solution of PIC polymers, and then once it reaches a certain temperature, the whole hydrogel just forms in one go. Sorry. And then the opposite, if you cool down, the hydrogel also disappears completely. Sorry. This is my time to say it's finished. So then I still had some slides about microrheology. Maybe I can just explain you very quickly that we want to be able to characterize the mechanical properties of the hydrogel. We need to follow the beads inside the hydrogel, and depending of the movement of these beads, we can retrieve the mechanical properties of the material. And to do that, we use a multiplane wide field setup. And the special thing about this multiplane setup is on the detection path. So we have this prisma, and the prisma, what it does is that it separates the emission that comes from the objective into 8 different paths that have a different length, which means that in practice you are imaging 8 different planes at the same time, okay? And this is what you get. So some molecules will be in focus on the first planes, and then you see that on the last planes they are out of focus, and the other way around with some molecules. So we are imaging at the same time 8 planes, and then we can add even a cylindrical lens to induce some aberration on the point spread function that can give you the information in 3D. And here you'll see the trajectory that we put a particle on the cover slide, and then we just make the stage make a certain movement, and we follow that by following the particle. So here you see the particle moving from in the different planes, and here you see the trajectory that we had retrieved from that movement. And then we use that to do tracking in 3D, and from that tracking in 3D we can calculate mean square displacements, and from the mean square displacements we can calculate diffusion coefficients, and from the diffusion coefficients we can calculate, in this case, the viscosity. If we have a hydrogel that has also an elastic component, we can also calculate the viscoelastic properties. And that was it. I think I made it nicely in time. So just like a slide showing actually a little bit of the two things, just about the cluttering pits, that we could use super resolution to really quantify this cluttering topology in metastatic cancer cells, and then that's how we could use fluorescent microscopy to characterize the structure of hydrogels. And then the thank you slide for everybody that is involved in the group and some collaborators. And thank you for your attention. And now I'll be very happy to answer some questions.
Thank you very much, Susana, for these very nice presentations you gave today. And well, now the forum is open for questions. Either we open the microphones to ask directly to Susana, or you can write down your questions in the chat, and then I will just pass questions to Susana. So please go ahead. If somebody has a question, please just go ahead.
You can also talk in Spanish. I can understand, but I will not answer in Spanish.
No, no worries, Susana. Bueno, si alguien tiene alguna pregunta, por favor, la pueden hacer directamente a Susana o la pueden escribir en el chat y yo se la puedo, yo se la haré a la doctora Susana para cualquier cosa, ¿vale? Okay. So Susana, well, just, I mean, I'm not in this field very some years ago, but the beginning when you were talking about these proteins, or let's say the markers, right? I was just wondering, how stable should they be to be used in this type of applications, at least for the biological part, you know, for the applications in cells, for instance?
Yeah, so in terms of stability of having the protein formed, they just need to form at 37 degrees, and I think most of the proteins, they do that. In terms of being able to use it for palm, that they can blink a lot, I have to say that in the beginning it was a little bit more of a challenge when people were first developing these proteins. But like Emao's protein is as bright as Atto dyes. So now the fluorescent proteins, that they are not a lot, but they are a few that are very good, and you can easily use them as you would use organic dyes. But there's still a lot of research ongoing on the development of new, more photostable fluorescent proteins, let me call it like that.
Okay, thank you, Susana. So we have also a question here from Elizabeth Rodriguez, and she question is the following. Related with DB scan, which are the parameters and how do you determine the correct values?
Yeah, so that was a little bit, I'm going to be completely honest there. So the parameters, let me try to find, I think I had an extra slide for that. So the DB scan is just a plugin on Fiji, I think, but you can also download it for MATLAB, and we do all our analysis on MATLAB, so we did that. And of course, that slide I don't have, so I'll go to the original slide. And what we did was we just try different types, so you can adjust two things, the minimum points and this distance. So you can say the points need to be at the minimum distance of, in this case, 0.55 to be considered to be in the same cluster. And you say that there is a cluster when they are at least, in our case, 17 molecules within this radius. How did we reach this? Was really a trial and error. We just first ran values between 0.3 and 0.7 and minimum points between 10 and 30. And we just look at the image and then we think, okay, this is, would we say that this is a cluster? Yeah, I would also say that this is a cluster. This maybe, this not, okay. And then like that, we just try to do it by eye, say, okay, this is a classification that we think is more correct, and then we just set it and we go with the same parameters. And some cells, maybe a different parameter would be better, but that we are not going to change between cells. We just took like that. And we do an additional step, that is to say, okay, we only keep the clusters that have more than 50. So we have, for it to be classified a cluster, it has to have a minimum of 17. And if we increase this value here, it starts merging clusters together. So we kept it at 17, but then we just say, for us to take it into consideration on our analysis, it needs to have at least 50. So it was very empirical way of finding these parameters.
Okay. And then we have the next question from Cecilia Garcia. So the question is, when you do the fitting, how do you determine what you are seeing is real? Because I suppose that you can do a fitting that eliminates important information, or a fitting that does not allow you to discriminate important information.
When you say fitting, it's the fitting of the single molecules with a 2D Gaussian. Is that the fitting that you are talking about? Or is there another fitting?
I suppose.
Yes.
Yeah, Cecilia say yes, yes.
There, it's kind of easier to do this discrimination, because when you are fitting with the 2D Gaussian, they are just the only parameter. Okay, there are two steps when you do the fitting with this 2D Gaussian. One is to find the molecules. What do you think can potentially be a molecule? And that's based on intensity, or sometimes in image gradient. This starts to be very image processing data. But so when you have a very bright spot and a background on the edge, there's a big difference. So that's a big gradient. So you need to say, okay, these bright spots have the potential to, they have the potential to be proteins, or to be, single molecules. And those molecules then you fit. And then you only discard those molecules if your Gaussian fit is very broad. And when does that happen? If the molecule is moving, then you don't have a very nice spherical 2D Gaussian, but it's a more elongated point spread function, and you want to discard it because the accuracy that you are detecting is not high enough. When you are discarding molecules, you don't discard the information because it is wrong, you just discard it because it's not accurate enough. So you can also calculate super resolution images, taking everything into consideration, but then your resolution that you have at the end is much lower. So you do this fitting control depending on the resolution that you want to achieve at the end. Does this kind of make sense? So if you don't care about the resolution that you achieve, you should say, for me 50 nanometers is also good. Just take all the molecules, and then you have all your data there. But if you say, but what I want to observe, it's really smaller than 50 nanometer, then you need to put your fitting parameters more stringent, and you discard the molecules that are not nicely fitted. But I don't think it's important information. In principle, it should just be molecules that are moving and you don't want to take into consideration.
Okay, Susana. So Elizabeth is also asking, well, it's more kind of a consultation, about time, about your experience with expansion microscopy.
Yeah. So that's somebody that checked my CV apparently. We do some expansion microscopy, but that is done a lot in collaboration with Johan Hofkens. So we had a few students that were working together, and I can tell you that our experience with expansion, it's a little bit a dual vibe. I cannot explain it in a different way. It has a lot of potential, and when it works, it's very nice. But you need to have some training on making it work. So you need to develop a feeling. And then we start talking about feelings in science. But you need to develop a feeling when you are handling your gel. Your gel is very fragile. So you need to have this, be very careful experimentally to be able to have it to work. But once it starts working, it works beautifully. But it's very hard to say, I'll send you the protocol and you can try it. If you are really interested, talk with Eduardo. He knows how to get to Belgium. And I would say just come to our lab for a couple of weeks and you can train with some people that are used to do it. And then you can have all the things that are not in the article on how you have to handle these things. And then it's very easy to do.
Okay. Thank you, Susana. Well, the girls, thank you for your replies. And just a final just a comment in here regarding what you have in this slide and what you were discussing about this DB scan and so on and so forth. Now that we have these machine learning hot topics and so on and so forth, will that be also useful for you guys? In this particular, in different ways?
Yeah, it is. And actually we do have some people that work on it and to do all this recognition, pattern recognition, but to be honest, for us it was just too much work to do it for this article. We still looked into it a little bit, but then we thought, no, this is going to take way too much time that we don't have.
Okay.
But definitely, and I think they are doing it a lot with the Fluorocode project, right? With the optical imaging of DNA. There they do a lot of this optical mapping, not optical mapping, sorry. Neural networks to try to identify what are the segments of DNA. But we honestly did thought of developing something like that for this project, to say that you can identify what is a lattice and what is a pit. We are, if there's anybody interested into looking at it, let me know. I can send you some data. No problem.
Yeah, I know that might be some interest on this, from both sides, But okay, anyhow, if somebody's interested, just please contact me and I will just give you the contact details of Susana, and then you can start chatting, after this seminar.
Yeah.
Okay, I think we are approaching the end, and I would like to thank you again, Susana, for accepting the invitation for this nice presentation, and thank you very much for to everybody. This is the last seminar of the year, and we are just closing everything with a very nice seminar. So, thank you again, Susana, y muchas gracias a todos. Muchas gracias a todos. Este es el último seminario del año y los esperamos el año siguiente. Thank you very much, guys. Have a nice weekend and have a nice Christmas party, Susana.
Okay, see you soon. Bye-bye.
Bye-bye.